Hi everyone, I'm Shuze Wang. I'm a fourth-year PhD student at University of Michigan. Today I'm going to talk about opposing gradients of retinoic acid and sonic hedgehog specify tonotopic identity in the mouse cochlea. Hearing is mediated by a specialized sensory epithelium, the organ of Corti, which is located in the cochlea of the inner ear. The organ of Corti is stretched along the apex to base, tonotopic axis of the cochlea. High frequencies are detected in the base, while low frequencies are detected in the apex. Generally, frequency tuning requires physical properties like bundle length, but also molecular components like ion channel to be organized in tonotopic gradients. Formation of gene expression gradients require tonotopic patterning of the cochlea. However, knowledge about this process is limited. Therefore, our aim is to identify the molecular mechanisms that confer spatial identity during cochlear duct extension. Before we discuss our findings, I would like to provide more background. During embryonic development of cochlea, a group of primordial cells at the base of the cochlea divides and thereby contributes to the elongation of the organ. Around embryonic day 12.5, E12.5, apical cells are born first, and during the following 48 hours, the primordial give rise to new cells that continuously elongate the cochlear duct until the final size is reached. A cross-section through the cochlear duct at E14.5 highlights the asymmetric organization of the cochlea at that stage. To resolve our goal, we proposed two hypotheses of cochlear duct extension. The first model is called time-space translation model. Time-space translation model is strictly controlled by time, which translates developmental time into spatial information. A mechanism like that has been discussed to establish expression pattern of Hox genes during Xenopus gastrulation. Specifically, the E12.5 apical cells are similar as the E14.5 apical cells, and the E12.5 basal cells are similar as the E14.5 mid-basal cells. The second model is called dynamic morphogen model, which morphogen-based signaling originating from the opposing end of the cochlear duct could potentially confer spatial information as well. Specifically, the E12.5 apical cells are similar as the E14.5 apical cells, while E12.5 basal cells are similar as the E14.5 basal cells. The major difference between these two models is whether the E12.5 basal cells are more similar as the E14.5 mid-basal cells or basal cells. To test the hypothesis, we generated single-cell RNA-seq data from the embryonic cochlear duct and spatially reconstructed the cochlear duct to investigate the gene expression gradient. First, we generated single-cell RNA-seq profiles from the developing cochlea at embryonic day 12.5, E12.5, and E14.5. To enrich for the target population, we dissected the inner ear from SOX2 GFP mouse line and separated the cochlea from the vestibular compartments. Cochlea samples were digested to single-cell level and subjected to the FACS. We sorted for the SOX2 GFP positive cells and proceeded with single-cell RNA-seq sequencing library preparation using 10x genomics platform. Each time points were represented by two independent replicates. After stringent quality control, a total of 1,700 cells were subjected for further analysis. We summarized the cell number for each time point and each replicates. Next, we identified the clusters and annotated cell types for each data respectively. By concentrating on cochlea duct cells, including cochlea floor and cochlea roof cells, we successfully reconstructed the 2D and 3D developing cochlea duct for E12.5 and E14.5 cochlea duct respectively. During development, the cochlea duct segregates into spatial domains asymmetrically arranged along the duct's circumsphere. Floor and roof segregates into medial and lateral compartments, and around E12.5, the zone of non-proliferating cells extend from apex towards base, while at E14.5, hair cells start to differentiate at the mid-basal cochlea duct and extends towards the apex and base. Here we generate a schematic representation of the asymmetric organization of the developing cochlea. To reconstruct the cochlea duct in 2D and 3D space, we assumed that the spatial domains of the developing cochlea are embedded in a continuum of gene expression. Based on this assumption, we resolved each cell's original position along the roof floor axis, medial lateral axis, and apex base axis in a PCA-based approach. The results were first projected onto the 2D scaled PCA plots. From the 2D projection, the x-axis distinguishes the cell types, and the y-axis represents the tonotopic axis with apex facing up. The 3D anatomy of the cochlea duct was reconstructed by projecting the 2D PCA onto a cylinder surface, whereby the head of cylinder was represented the longitudinal tonotopic axis of the developing cochlea duct. From the 3D cylinder plots, we can observe that the height of E14.5 cylinder is higher than the E12.5 cylinder, which simulates the cochlea duct extension process. For better visualization, we generated a circular projection by flattening the apex base axis. The circular projection resolves the asymmetric organization of the cochlea. Within the circular plots, the roof cells are at the top, while the floor cells locate at the bottom of the circular plots. From left to right, it represents medial to lateral axis, with medial on the left side and lateral on the right side. Additionally, the apical cells are close to the inner circle, while the basal cells are close to the outer circle. Overall, the circular projection specifies the three major axes of the cochlea, including roof, floor axis, medial lateral axis, and apex base axis. To validate our 2D reconstruction, marker genes were used to project onto these donut circular plots. Take E14.5 dataset as an example. OC90 and LUM characterize the roof and floor cells. The in situ hybridization validates our 2D reconstruction. From the image, we can observe the OC90 is highly expressed in cochlea roof, and the LUM is highly expressed in the cochlea floor. Similarly, FGF10 and BMP4 distinguish the medial and lateral side of the cochlea. And FST and MKI67 are the marker genes represent apex and base. After estimating each cell's relative position within the developing cochlea duct, our next aim is to understand the molecular mechanism that characterizes the spatial identity during cochlea duct extension. Just want to remind, we want to know either time controls the tonotopic identity or morphogen regulates the spatial identity during cochlea development. Therefore, we propose these two hypotheses, time-space translation model and the dynamic morphogen model. To test our hypothesis, we focused on the cochlear floor cells and first identified gradually expressed genes along the tonotopic axis using KL divergence. Here we screened two candidate genes, HMGA2 and HMGB2. We ordered the cells along the x-axis from base to apex. HMGA2 is a gradually expressed gene with increasing level from base to apex, and HMGB2 has the opposite gradient along the tonotopic axis. Additionally, we validated the gradually expressed genes using FISH. Next, we leveraged the gradually expressed genes as feature to test our hypothesis. We aggregated 10 cells as a metacell and calculated the Euclidean distance for each pairwise metacell between E12.5 and E14.5 data. Next, we converted the Euclidean distance matrix to similarity matrix using Gaussian kernel. Heat map were used for visualization. Both columns and rows are metacells ordered along the tonotopic axis. Metacells from the E12.5 apical cells were most similar to the E14.5 apical cells, and the E12.5 basal cells are aligned with E14.5 basal cells, although a lot of cells has been added to the basal end. Therefore, our findings support the dynamic morphogen model. To test the robustness of our finding, various parameters that were used to generate the similarity matrix were systematically tested, and overall, they all consistent with our previous finding. A quick summarization here. Our finding supports the dynamic morphogen model rather than the time-space translation model. Our next question is, what are the morphogens involved in tonotopic patterning? According to the previous publication, in the mouse cochlea, constitutive activation of SHH, sonic hedgehog signaling, causes an expansion of apical genes and a loss of basal genes. In the absence of SHH signaling from the spiral ganglion, the regional identity were required normally and the tonotopic gene expression gradient didn't change in the shortened cochlea. This indicates that SHH mediates apical identity of the cochlea in mice. From our differential goal-term pathway analysis, we find retinoic acid signaling, RA signaling, may pattern the basal identity as well. And therefore, we hypothesize that RA and SHH form an equilibrium of two opposing morphogen gradients, which jointly control the tonotopic patterning of the cochlea duct. Our next aim is to test the hypothesis by perturbing the RA and SHH morphogens. E14.5 explants were cultured for 72 hours, and changes in HMGA2 expression were quantified using FISH. HMGA2 is a reporter gene. In an effort to focus on the analysis on cell types contributing to the development of the organ of Corti, explants were co-stained with an antibody SOX2 to highlight the prosensory domain region. First, differential expression of HMGA2 along the tonotopic axis was confirmed after 72-hour culture for the DMSO control. Next, explants were cultured for 72 hours and supplemented with ectopic RA, which resulted in significantly reduction of HMGA2 expression in the apical compartment. Since RA patterns the basal compartment, the ectopic RA made the apical cells more similar as the basal cells, which was expected. Conversely, culture with RA receptor antagonist AGN significantly increased the HMGA2 transcript level at the base. Activation of SHH using smooth agonist SAG resulted in significantly increase in HMGA2 expression in the basal compartment, which was similar to the observation made after blocking RA signaling transduction. On the other hand, supplementation with SANT1 antagonists that directly inhibit SHH pathway by binding to the SMU receptor significantly decrease the HMGA2 expression, comparable to the levels seen after application of ectopic RA. In summary, the occurrence of mirrored phenotypes strongly suggests that RA and SHH jointly pattern the tonotopic identity in the developing cochlea. In summary, we reconstructed the 2D and 3D developing cochlea from single-cell RNA-seq data. From the hypothesis testing, we predicted that the morphogen encodes the spatial information instead of time. And also, we identified RA signaling as a likely candidate to confer the basal identity. Finally, the mirrored phenotype suggests an equilibrium of RA and SHH controls the tonotopic identity during cochlear duct extension. And here, I would love to thank everyone in our lab, especially SciCut, who did all of the wet lab experiment to support our results. And thank you so much for listening. I'm happy to take any questions.